Hello everyone, welcome to the today's lecture. In today's lecture we are going to discuss about gram negative bacteria cell wall. In earlier lecture we discussed about the gram positive bacteria cell wall. Okay, so now we are moving on the gram negative bacteria. So in this lecture we are discussing the gram negative bacteria cell wall and what are the component and how it is different from the gram positive bacteria. Okay, so just look at the structure of gram negative bacteria cell wall. In this structure, if you are looking the structure of gram negative bacteria cell wall, what we found, just look at this portion is basically related with the cell membrane. Okay, so this portion is not a part of cell wall. Now then, what is the part of cell wall? So this is periplasmic space. Okay, this portion is known as a periplasmic space, and now just look the green line. Okay, so this green line is known as a peptidoglycan. Okay, then again this area is known as the outer membrane. So this whole area is known as the outer membrane. So in the structure of gram negative bacteria cell wall, we can found three structures. One is the outer membrane. Okay, then again the space between peptidoglycan and outer membrane is known as a periplasmic space, and again the space between peptidoglycan and cell membrane or plasma membrane is known as a periplasmic space. So here we found two places. So here in the gram negative bacteria we found periplasmic space between two places. Okay, the first one is between the outer membrane and the peptidoglycan, and the second one is between the cell membrane and the peptidoglycan. Okay, so I'm just writing over here what are the component of gram negative bacteria cell wall. Okay, so the first here, just look at this gram negative, okay, cell wall. It has a three component. Outermost we can call it as a outer membrane. Okay, and then the second one is peptidoglycan layer, and the third one is periplasmic space. Clear everyone? So in earlier lecture of gram positive bacteria we discussed the structure of peptidoglycan. So first I am considering here the structure of peptidoglycan. Okay, but before discussing the peptidoglycan structure, I am just describing you that what periplasmic space contain. So just look at here, this is the portion of periplasmic space and this is the portion of periplasmic space. So here we found a gel-like fluid. So this gel-like fluid is found in the periplasmic space, and just remember that periplasmic space is too large in gram negative bacteria and it is very small in gram positive bacteria, or we can consider it as a negligible in gram positive bacteria. Okay, that's why in so many books the authors written that periplasmic space is absent in gram positive bacteria, and we can found a big layer of periplasmic. We can found a high amount of periplasmic space in gram negative bacteria. Now just looking on the second, that is peptidoglycan. Okay, so just look at the peptidoglycan, how much peptidoglycan layer is in the gram negative bacteria. So in gram negative bacteria the peptidoglycan layer is only 1 to 3 layer we can found, and the thickness of this layer is very short. We can say 2 to 3 nanometer range we can found in the gram negative bacteria. So the layer is very thin. The layer is very thin in comparison to gram positive bacteria. So in gram negative bacteria we found the thin layer. However, in the gram positive bacteria we found a bigger or thicker layer in them. Here we found very thin layer. Just look at the 2 to 3 nanometer range. Okay, but in the gram positive bacteria the layer is very thick. Okay, so that is the big difference. On this difference we differentiate them in gram positive and gram negative bacteria on the basis of staining. Okay, so just looking the structure of peptidoglycan, how it is differ from gram-positive bacteria. So in earlier class of gram-positive bacteria, I discussed you that peptidoglycan is a polymer. Peptidoglycan is a polymer. It contains NAM, NAG, NAM, NAC, NAM and NAG alternatively. So these are the compound which found in this polymer alternatively. What is the full form of NAM? So NAM is basically what? It is a N-acetyl muramic acid. Okay, and what is NAG? So it is a N-acetyl glucosamine. Okay, so just look at the structure. In NAM and NAG structure, they are alternatively arranged, and NAM having a tetrapeptide connection. So NAM is connected with the tetrapeptide. So this tetrapeptide is made up of 4 amino acid. Okay, so I am showing you this structure by the help of model, which I earlier show in the gram-positive bacteria, but there are some differences. Just look at the model. Now just look at the structure. Similarly, this first rod is basically made up of NAM and NAG. Okay, so these are some various lines of NAM and NAG. Okay, so just look at the structure. This one is basically, this yellow color ball is basically our NAG. Okay, so I am starting with here. So just look at the various NAM and NAG structures. Red color ball is NAM, then NAG, NAM and NAG, NAM and NAG, NAM and NAG. Okay, again similarly NAM and NAG and alternatively arranged NAM and NAG, alternatively arranged NAM and NAG and alternatively arranged NAM and NAG. Okay, here I show you that NAM contains 4 tetrapeptide, okay, which we earlier discussed in gram-positive bacteria. So these 4 tetrapeptides are, the yellow color ball is basically a L-alanine, and then second blue color ball, that is known as a D-glucosamine. You can consider it as a D-glucosamine. Here the third amino acid, which is changed from the gram-positive bacteria. In gram-positive bacteria, it is a L-lysine, but here it is, I am showing you with the pink shiny color structure, that is basically a D-Meso diaminopimelic acid. Remember, it is what? D-Meso diaminopimelic acid. Okay, and then fourth amino acid is same, that is a D-Alanine. Okay, again similar here in the second row we have a what? Again same tetrapeptide. Okay, this is the tetrapeptide. So same amino acid are associated with this, and the third number is change. Okay, so now what are the difference? In the second row, again there is a same connection with them. Okay, so now just look at how they are connected with each other. Okay, so again there is a third amino acid of tetrapeptide is connected with the terminal amino acid of other row. Okay, just look at the structure, how these tetrapeptides are connected. Here the tetrapeptides are connected with each other by the help of a covalent bond. Here they do not making a pentaglycine bridge. Here they are not making the pentaglycine bridge. The same pentaglycine bridge is made up between the third amino acid and the terminal amino acid in gram-positive bacteria. Okay, but in gram-negative bacteria, we do not found pentaglycine bridge. Instead of that, we found only a covalent bond. So they simply connected with the single covalent bond. Okay, so this is the covalent bond between them. Clear everyone? So this is the big difference between gram-positive and gram-negative bacteria. So there are two differences. Okay, the first one is the third amino acid is changed from gram-positive bacteria, and the second one is the pentaglycine bridge, which is absent in gram-negative bacteria cell wall. Okay, so remember this. On this, examiner asks questions so many times. So this is the structure of peptidoglycan in gram-negative bacteria, how it is arranged. Now I am discussing the structure of outer membrane. Okay, so now just look at the structure of outer membrane. Here in outer membrane we found two type of parts. The first one is outer leaflet. Okay, outer leaflet, and the second one is inner leaflet. Okay, outer leaflet is made up of 3 portions. Okay, what are these 3 portions? The first one is lipid A. Okay, first one is lipid A, which is also known as an endotoxin, toxin. Then second one is core polysaccharide. Here everyone. And the third one is O antigen. Third. Now if you are looking the structure of this lipid A. So what is the structure of lipid A? So lipid A is made up of 2 glucosamine unit, 2 glucosamine unit, okay, and these 2 glucosamine unit basically made up of 3 fatty acid plus phosphate. So each of 2 glucosamine unit is made up of 3 fatty acid plus phosphate. So total there are 3 to 8 unit long it is. Okay, so lipid A is 3 to 8 unit long and it contains 3 fatty acid plus phosphate. So total 8 unit we can found in the lipid A molecule. So now just look at the lipid A. What are the function of lipid A? Or basically it produces various endotoxins. Okay, so it has a toxic activity. So what are those toxic activity of lipid A? So these are basically a lethal action. Okay, it produces lethal action. The second one is it can also induce antigenicity. Okay, then third one is a toxic we can say, and it is also producing mutagenic reaction. Then fourth one is tissue necrosis and pyrogenicity caused by this endotoxin of various gram negative bacteria, as well as the B cell mitogenicity is also caused by gram negative bacteria endotoxin. Then anti-complementary activity is also produced by lipid A endotoxin. So these are various toxic activity produced by lipid A. That's why gram negative bacteria are more dangerous than gram positive bacteria. Remember, due to just lipid A or endotoxin we can say that presence of this lipid A can make gram negative bacteria more complex as well as more toxic to the other species. Okay, then core polysaccharide. So core polysaccharide is basically 10 to 12 sugar moiety contain. Clear? So it contains 10 to 12 sugar moieties. Okay, and then just look at the structure. This is our lipid A structure. This is what a lipid A, and then just look the structure of this black color structure is known as a core polysaccharide. Okay, and then this structure, the thread like structure, is known as O antigen. Okay, so now just looking the structure of O antigen. So O antigen is basically this thread like structure and it is made up of several sugar moiety. So I can write here several sugar moiety it contain. Okay, and we found it on the surface. That's why we are calling it as a surface antigen. As it working as an antigen, so it can induce antibody forming reaction. So it can induce what? Antibody forming reactions. Okay, clear everyone? So these are the structures of outer leaflet. Just looking the structure of inner leaflet. So this is the structure of gram negative bacteria outer. Okay, the structure is basically made up of phospholipid. This structure is basically made up of phospholipid. Okay, this phospholipid is very much similar with the phospholipid which we found in the cell membrane, or we can say plasma membrane. Okay, in the inner leaflet, in this area, what we found? We found outer membrane protein. What we found? We found outer membrane protein. Okay, if the 3 outer proteins are making the cluster, okay, if the 3 proteins are making the cluster, then what they are making? They are making a Porin channel. What they are making? They are making a Porin channel. Okay, and what is the function of Porin channel? Just look at the structure of Porin channel. This is the structure of Porin channel. Here 3 outer membrane protein are making a cluster and they are making a Porin. Here also 3 outer membrane proteins are making a cluster. Okay, clear everyone? So what is the function of Porin channel? This Porin channel basically what they do, they allow 600 to 700 Dalton molecular weight compound to pass from the cell wall. Okay, remember this Porin channel are allowing the 600 to 700 Dalton molecular weight compound to pass from the cell wall from inside and from outside, okay, both the way. So we can found so many of the Porin channel, within the gram-negative bacteria cell wall. Remember this, in gram-positive bacteria, very few species containing Porin channel. That's why we are considering in the gram-positive bacteria that Porin channel are absent. Most of the gram-negative bacteria containing so many of the Porin channel. So you can consider here there are so many Porin channel, okay. So that is the important work. And if the compound like vitamin B having a higher molecular weight, then how such kind of compound can transfer from the cell wall, then in such kind of compound gram-negative bacteria needed a carrier molecule. So by the help of carrier molecule, gram-negative bacteria can transfer such kind of compound, okay. So this is the structure of inner leaflet. One important point we found in the outer leaflet is what? Bronze lipoprotein, okay. So bronze lipoprotein also found in gram-negative bacteria, but not in the all species, only few species containing bronze lipoproteins, okay. So bronze lipoprotein is attaching the inner leaflet with the peptidoglycan, okay. Bronze lipoprotein is the connection, or we can say attachment, between the inner leaflet lipoproteins and the peptidoglycans, okay. Then one important structure we found outside the outer membrane and which is associated with the outer membrane. What is this structure? The structure associated with the outer membrane is S layer. What we are calling it? We are calling it as a S layer. So just look at the structure of S layer. This blue color line here I show is the outermost layer, and we found only in the gram-negative bacteria, okay. So this S layer we found in the gram-negative bacteria as the outermost layer, okay. Now generally what scientists think, or students of the science background think, how can we demonstrate the cell wall in the lab? This is the question with us. So what are the various methods to demonstrate the cell wall in the lab? Cell wall demonstration. So what are the various methods of cell wall demonstration? Just look at the various methods of cell wall demonstration. The first one is plasmalysis. So what is plasmalysis? Plasmalysis is what? When we put gram-positive or gram-negative bacteria in the hypertonic or hypotonic solution, then there is a possibility of shrinkage or bursting. Two kind of reaction we can found. So by this way we are demonstrating the plasmalysis of bacterial cell wall. The second one is what? Cell dissection. Okay, this is the second method. Then the third method is what? The third method is differential staining. Okay, so how we are doing the differential staining? I am just going to discuss in next few lectures. So with the help of staining, what we do, we stain the bacteria with the help of dye. And in the cell wall, we found a major difference between the peptidoglycan layers. Okay, so due to the difference between the peptidoglycan layer, there is a difference in the staining. Okay, so by the help of differential staining, we are able to see the cell wall demonstration. Okay, next one is by the help of electron microscope. Okay, and then the fifth one is reaction with specific antibody. So by the help of reaction with the specific antibody, we are able to demonstrate the cell wall. Why I discuss this cell wall demonstration? Because in CSIR, NAT, GPAT and other NEET PG competitive examination, examiner asks one question: what are the method of cell wall demonstration? So these are the various method of cell wall demonstration. Okay everyone. So in this lecture, I discuss about the structure of gram-negative bacteria cell wall, and I show you some differences. So what are those differences between the first difference in the peptidoglycan layer? Peptidoglycan layer is thin in the gram negative bacteria and gram positive bacteria it is a thicker. Second difference in the third amino acid in the gram negative bacteria is D-diaminopimelic acid in tetrapeptide, and in gram positive bacteria it is a lysine. Okay, it is a what? Lysine. And here in the gram negative bacteria we only found the covalent bond, and in gram positive bacteria we found what? Pentaglycine bridge. The another point is what? We do not found any kind of thicoic acid or lipothicoic acid in gram negative bacteria. However, both acid are present in gram positive bacteria. Okay. The another important thing is what? Periplasmic space. Periplasmic space is not present in gram positive bacteria, but in gram negative bacteria we found lot of amount of periplasmic space. Bronze lipoprotein also present in gram negative bacteria, as well as lipopolysaccharide LPS. Lipopolysaccharide is present, lipid A is present, which is not present in gram positive bacteria. Okay, so these are some major differences between gram positive and gram negative bacteria cell wall. Okay, so I hope you are able to understand these points. Here I discuss some questions with you, so answer these questions in the comment box, and like the video, share the video and subscribe the channel.
Hello everyone, discussing the question number 1. Cell wall of gram negative bacteria contain A. Peptidoglycan, B. Periplasmic space, C. Lipopolysaccharide, and D. all are correct. Question number 2. True about peptidoglycan of gram negative bacteria cell wall. A. 1 to 2 layer and 2 nanometer thick. B. 100 to 200 layer and 20 nanometer thick. C. 30 to 50 layer and 200 nanometer thick. D. all are correct. Question number 3. True about gram negative bacteria cell wall. A. Meso diaminopimelic acid is present at third position of tetrapeptide side chain ascent from NAM molecule. B. Pentaglycine bridge is absent. C. The tetrapeptide side chain are directly linked to each other by the covalent linkage between D-Alanine of one chain with Meso diaminopimelic acid of the adjacent chain. D. all are correct. Question number 4. True statement for cell wall of gram negative bacteria. A. It contain outer membrane made up of phospholipid. B. Thicoic acid absent. C. Both are correct. D. None are correct. Question number 5. True statement for lipopolysaccharide of cell wall of gram negative bacteria. A. It contain lipid A or endotoxin. B. It contain core polysaccharide. C. It contain O side chain O antigen. D. all are true.